Hello, my name is Carrie Gibbons, and I am the Environmental Microbiology Team Lead at the U.S. Geological Survey's Upper Midwest Water Science Center. I am based in Lansing, Michigan, where we have an environmental microbiology lab. I am part of an interdisciplinary, multi-center USGS collaborative effort funded by the U.S. Environmental Protection Agency's Great Lakes Restoration Initiative to characterize harmful algal blooms in the Great Lakes. Our study site is in Lake Erie, specifically the Western Lake Erie Basin. The Western Lake Erie Basin is plagued annually by recurring Cyanobacteria blooms that produce toxins known as cyanotoxins. These blooms are frequently referred to as harmful algal blooms, or HABs. The Western Lake Erie bloom varies annually in size, location, duration, and toxicity. Typically, it starts in the western corner near the Maumee River mouth in Maumee Bay and extends east along the southern shoreline of Sandusky Bay and north along the western shoreline. More recently, there has also been a central basin bloom. Blooms typically occur in summer months during late July through September and can produce cyanotoxins such as microcystin. These blooms cause recreational and drinking water concerns. Lake Erie is a source of drinking water, and several coastal cities, such as Toledo, have drinking water intakes in bloom areas. In 2014, microcystin producing cyanobacteria infiltrated the Toledo drinking water intake in the water plant and resulted in a 2-day shutdown that affected over 500,000 people. Many of these coastal cities, such as Toledo, have deployed sensors and developed new technology to try to detect microcystin producing cyanobacteria in the source water, and then also find ways to remove it from the source and finished drinking water. USGS Lake Erie HABs research began in 2013 and expanded in 2014 with an ecosystem approach monitoring study to understand the role that biotic and abiotic factors, not just nutrients, have on harmful algal blooms. We incorporated what we knew from the literature in our own current studies and then tried to ask some overarching big picture questions, such as, are there forms of phosphorus, nitrogen, carbon, and other trace elements that impact the formation and cessation of harmful algal blooms? Another one was, do we see cyanobacteria in the sediment, and are these the same cyanobacteria groups that we see within the water column? Is it possible that cyanobacteria within the sediment might be a source or a seed for the following year's bloom? For sample collection, we collect nutrients and water quality constituents throughout the lake. We also collected lake bed sediment for toxin and microbial analysis. Sediment was collected from April to May prior to blooms at these sites noted with either orange dots or brown circles. Many of these sites were located near river mouths, such as 15D, which was located at the Detroit River mouth, 1M, which is at the Maumee River mouth, and WQ2, which is at the River Raisin River mouth. The blue dots indicate sites where we collected water samples from June to October. The main goals of our multi-year study were to better understand the spatio-temporal life cycle of harmful algal blooms, determine toxins associated with HABs in Lake Erie, quantify relative abundance of genes associated with cyanobacteria and cyanotoxin production, characterize microbial communities associated with algal bloom formation and toxin production. And the main question we were trying to answer was what abiotic and biotic factors influence HABs and their toxins, and how do blooms influence ecosystem services. For the purpose of today's talk, I'm going to focus on our work characterizing microbial communities and the water column samples collecting during field years 2014 and 2016. Our work aimed to characterize the bacterial community in the sediment in the water column. We also wanted to assess microbial community succession in the water column prior to, during, and after blooms, and how the microbial succession varied with toxin genes and phytoplankton community profiles. Our main goal was to understand how bacterial community structure is related to algal bloom formation and toxin production. To do that, we filtered water samples in the field, extracted the DNA in the lab, and sent the DNA for sequencing on the aluminum MySeq. We use the mother bioinformatics pipeline to understand who's there in our microbial community composition within our samples. Knowing who's there provides key information on what microbes are naturally present and how the community shifts with changes in the environment, including increased nutrient or contaminants, which is what we see with harmful algal blooms. Now a little bit about our results. For the results today, I'm focusing on water column samples collected at WQ1, WQ2, and WQ5 from the years 2014 through 2016. It is important to consider that WQ1, WQ2, and WQ5 are likely influenced differentially by riverine influences from the Maumee River, River Raisin, and Detroit River, respectively. 11 bacteria phyla contribute over 1% of the total sequencing reads from our MySeq sequencing runs. Of those 11 phyla, there is considerable variation in the contributions of bacteria within Actinobacteria, dark blue bar, Bacteroides, light green bar, Cyanobacteria, red bar, and Proteobacteria, yellow bar. You will also note that there is considerable variability among the 2014, 2015, 2016 season at all three sites, and between the sites over all three seasons. One of the key things to note here is that Cyanobacteria frequently contributes a large portion of the sequencing reads at WQ1 and WQ2, but less so at WQ5. This may indicate that WQ1 and WQ2 sites are more influenced by the harmful algal bloom in Lake Erie, and we may be able to tease apart what is different between the abiotic and biotic factors influencing WQ1, WQ2, and WQ5, and how this may translate to the Cyanobacteria portion of the microbial community. Here we look closer at the Cyanobacterial purple bar community dynamics at our three sites, WQ1, WQ2, and WQ5, over the 2014 through 2016 seasons. Within the Cyanobacteria, we focus on Limnothrix, Microcystis, teal bar, Planktothrix, and Synechococcus, yellow bar, OTUs. Once again, these are the percent of total sequence reads for the sequencing library and suggest relative abundance of bacterial groups as binned into OTUs. WQ1 is our site most influenced by HABs dynamics, and we see there are numerous states in 2014 when the percent contribution of Cyanobacteria was over 40%. Prior to the visible bloom, Synechococcus dominated the Cyanobacteria community, but in late July we see that community flip to Microcystis dominated, and we have resulting cyanotoxin production. We see something similar occur at WQ2 in 2014. However, WQ5 is less influenced by Microcystis and remains Synechococcus dominated through much of 2014. At WQ1, we do see a second bloom and resulting increase in Microcystis contribution in late September. And indeed, we did have a second smaller bloom around the state, which did produce some toxins, albeit less relative to the early August bloom. In 2015, we see a different pattern emerging at WQ1, with Cyanobacteria not becoming dominant in the microbial community until we have that initial algal bloom in Microcystis, increase in Microcystis in late July, early August. We do have toxin production then with that bloom, and then after that, the Microcystis and the Cyanobacteria contribution dissipate as the algal bloom goes away. We do see a strong cyanobacterial signal in 2015 at WQ5, but this is mainly Synechococcus dominated, except for the mid-August bloom, with the strong Microcystis contribution that we also see at WQ1 and WQ2. In 2016, we see contribution from Cyanobacteria, but this is mainly from Synechococcus, and notably the algal bloom in 2016 was much more moderate in comparison to 2014, which was highly toxic, and 2015, which spanned a large spatial area. So the 2016 bloom was smaller, less toxic, less Microcystis production. One of our main goals with our microbial community data was to look at how non-cyanobacteria bacterial groups, the heterotrophic bacteria, varied with cyanobacteria groups and HABs dynamics. You might think of this as the algal bloom microbiome, or as some researchers, most recently in Pearl et al. 2020, have coined it, the Microcystis interactome. We know that heterotrophic bacteria can influence the water environment and also be influenced by the water environment, especially with the presence of increased nutrients or the formation of algal blooms. We can think of this as a synergistic relationship between the heterotrophic bacteria and the cyanobacteria, and we are most interested in what heterotrophs may be benefiting from the formation and proliferation of algal blooms or the senescence of algal blooms in decaying cyanobacteria biomass. This particular figure details the top 20 OTUs in the water column in WQ1 during the 2014 and 2016 field seasons. Over all of our seasons in the WQ1, WQ2, and WQ5 water column samples, these top 20 OTUs contributed 62% of total sequencing reads within the sequencing library. The two cyanobacteria OTUs are ubiquitous in almost all of our water samples in our Synechococcus, yellow bar, and Microcystis, orange bar. Additionally, these two same OTUs were found in almost all Lake Erie bed sediment samples, suggesting a potential for cyanobacteria sink and source within the bed sediment and potentials for next year's bloom. From published literature, we know a little bit about several of our top 20 OTUs. Several of them are frequently detected in freshwater environments and often associated with algal blooms. Limnohabitans, dark blue bar, has been found to be associated with spring phytoplankton blooms and also decaying cyanoalgal biomass senescence. The Cyanobacteria groups CL500-29 in the HCl clade, noted by the light gray, light green, and light blue bars, have a growth association with blooms, but literature seems to still debate whether these groups are enhanced or decreased by bloom growth. Microbacteriaceae, the bright teal bar, is a frequent dominant bacterial group during documented blooms, and our data seem to agree with previous published results, as this group does tend to increase in relative abundance when we see the Microcystis bloom. Sedimentbacterium, which is dark gray bar, is often frequently associated with algal blooms and has previously found to be attached to algal cells. Our results show that there is quite a few relationships that need further teasing out in. One of our main goals with our microbial community data was to look at how non-cyanobacteria bacterial groups varied with cyanobacteria groups and HABs dynamics. You might think of this as the algal bloom microbiome, or some researchers, most recently in Pearl et al. 2020, have coined it the Microcystis interactome. These non-cyanobacteria bacteria groups, or the heterotrophic bacteria, can influence the water environment and also be influenced by the water environment, especially with increased nutrients and the formation of algal blooms. We are quite interested in the synergistic relationship between these heterotrophs and the cyanobacteria portion of the microbial community. We're specifically interested in which groups seem to have a positive relationship with the initiation, formation, and proliferation of algal blooms, and also the senescence of algal blooms and cyanobacteria die off. This particular figure details the top 20 OTUs in the water column in WQ1 during the 2014 and 2016 field seasons. These top OTUs ended up contributing approximately 62% of total sequencing reads across all seasons in water column samples from WQ1, WQ2, and WQ5. Two cyanobacteria OTUs were ubiquitous in almost all of our water samples in our Synechococcus, yellow bar, and Microcystis, orange bar. These same two OTUs were also found in Lake Erie bed sediments, suggesting a potential source or sink for cyanobacteria cells. From the published literature, we know a little bit about our top 20 OTUs. We know that they're frequently detected in freshwater environments, and several of them have close association with algal blooms. For example, limnohabitans with the dark blue bar has been found to be associated with spring phytoplankton blooms and also decaying cyanoalgal biomass, what we call senescence. Sedimentabacterium, dark gray bar, is often frequently associated with algal blooms and has previously been found to be attached to algal cells. Microbacteriaceae, the bright teal bar, is a frequent dominant bacteria group during blooms, and our data would agree with this previous result as we see increases in the relative abundance of this heterotrophic bacteria with increases in Microcystis community. The Cyanobacteria groups, which are denoted as the light gray, light green, and light blue bars, also seem to have a growth association with blooms. But like the literature, our results do not seem to indicate whether these groups are enhanced or decreased by bloom formation. Our early results indicate interesting trends that need further analysis, which we will be completing in the upcoming months. This is an MDS ordination of the Sorensen distance matrix, and we are essentially looking at an ordination of observed species richness. Bloom influence communities in WQ1 and WQ2 ordinate in the right bottom corner and away from most of those samples of WQ5. We find statistically significant clustering in the ordinations and a significant difference in the community structure between that of WQ1 and WQ5 and also that of WQ2 and WQ5. When we look at an NMDS plot of an ordination of the Bray-Curtis distance matrix, we are looking at an ordination of observed species richness and abundance of those species. The abundance of Microcystis versus Synechococcus OTUs influences some of the spatial ordination. 2014 Microcystis bloom samples plot to the right, as do those from 2015. However, much of 2015 was Synechococcus dominated, and those samples plot in the left bottom corner. We find statistically significant clustering in the ordinations between WQ1 and WQ5, and also that of WQ2 and WQ5. With both community structure, who's there, and membership considering relative abundance, we see a significant relationship between Microcystis production and that of our community structure. We also see a statistically significant difference between those samples occurring during Microcystis blooms and those occurring before and after the Microcystis blooms. We will be delving into this further and really looking at what abiotic and biotic factors may be driving these microbial patterns. This has been a very high-level synthesis of our work. In the coming months, we will be synthesizing the 2014 through 2018 Western Lake Erie Basin microbial community dynamics in the water column and sediment. We will specifically be looking at the relation of the microbial community to measured water quality constituents, toxin production, cyanobacteria and cyanotoxin genes, phytoplankton biovolume, and other variables. We hope to further understand the response of dominant and rare members of the microbial community and their synergistic relationships. We also have environmental metagenome data from the 2015 and 2016 years, and we look forward to looking at that in relationship to potential functional dynamics of our microbial community. during harmful algal bloom formation and proliferation in Lake Erie. And with that, I'd like to conclude my presentation. Thank you for attending. If you have questions, please feel free to ask me during the question and answer session or email me at cgivens@usgs.gov.